Okay, let's unpack this. Cancer treatment, I mean, it's seen incredible breakthroughs, especially with something called immune checkpoint blockade, or ICB.
Right.
It's been a true game changer for so many people, offering a real renewed fight against cancer. But here's the complex truth. It doesn't work for everyone.
No, it doesn't.
Why is that? And what are the hidden costs of essentially unleashing our body's own powerful immune system?
Well, what's fascinating here, I think, is that we're really talking about a fundamental shift, teaching your body's own defense system to recognize and attack cancer. It's about recalibrating.
Recalibrating, I like that.
So today, our mission is to really take a deep dive into the hallmarks, the key features that explain why some people respond brilliantly to these treatments, others unfortunately resist, and some experience pretty significant side effects.
And we're drawing on some serious research here, right?
Yeah.
Experts like Morad, Helmink, Sharma, Wargo.
Exactly. Some of the leading work in the field, trying to piece this all together to give you, the listener, a clearer picture of this, well, incredibly intricate science.
Precisely. Think of it maybe like peeling back the layers of an onion to understand the delicate dance between your body and these revolutionary therapies.
That's a good analogy.
You're going to walk away, hopefully, with some surprising facts and a clearer, really in-depth understanding of this complex science without feeling, totally overwhelmed. Let's try and make every piece of this puzzle make sense for you.
Let's do it.
So, to start us off, what exactly are these immune checkpoints we keep hearing about? And why do cancer cells seem to, well, love them so much?
Okay, so our immune system is incredibly powerful, right?
Mm-hmm.
But it also has built-in brakes. They're absolutely crucial.
Brakes, like on a car.
Sort of, yeah.
Yeah.
They're there to prevent the immune system from attacking healthy tissue, preventing autoimmune problems after it's dealt with an infection or another threat.
Makes sense. You need to stop it from going wild.
Exactly. But cancer, in its really cunning way, has learned to essentially push down on these very same brakes.
Ah, okay.
It basically tells our anti-tumor T cells, the soldiers of the immune system, to just stand down, leave it alone, like putting up a do not disturb sign.
That's a perfect way to put it, a biological do not disturb. So the whole goal of immune checkpoint blockade, ICB, is to release those brakes, to let the T cells hit the gas again.
Precisely. To take the foot off the brake,
Huh.
or maybe even cut the brake line, depending on the target.
Okay, so where did scientists begin this journey? Which brake did they try to cut first?
Well, the first major success story, the one that really kicked this field into high gear, was targeting a molecule called CTLA4, cytotoxic T lymphocyte-associated protein 4.
CTLA4. Right, let's break that down. What's its natural role? How does it actually hit the brakes in the body?
So think about T cell activation. That's like starting an engine. You need a key, that's the antigen, the thing the T cell recognizes. But you also need to press the gas pedal, that's a co-stimulatory signal, often through a molecule called CD28.
Key and gas pedal. Got it.
Right. Now, CTLA4, which is also on the T cell surface, acts like a competing brake pedal. It binds to the same signals that CD28 binds to, but it binds much, much more tightly. Higher affinity.
Oh, so it outcompetes the gas pedal.
Exactly. It effectively wins the competition, pushes CD28 aside, and slams on the brakes, actively suppressing the T cell's activation. This is particularly important for regulating those CD4 plus helper T cells, kind of the quarterbacks, and it also boosts the activity of suppressive cells called TREGs, regulatory T cells.
TREGs. They sound important.
They are. They help maintain tolerance normally, but in cancer, they help shield the tumor. So CTLA4 creates this overall environment where the immune system is dampened, allowing tumors to grow, effectively ignored.
Okay, so by blocking CTLA4 with an antibody, a specific drug, we're essentially cutting that brake line, stopping it from binding and suppressing the T cells. What happened when scientists first tried this in patients? This sounds huge.
It was huge. The preclinical studies in mice looked really promising, showing enhanced, long-lasting anti-tumor responses. Then, clinically, humanized anti-CTLA4 antibodies like ipilimumab were developed and eventually approved for advanced melanoma.
I remember the buzz around ipilimumab.
Absolutely. It was revolutionary because at that time, there really weren't other therapeutic options that could significantly increase survival for those patients. Ipilimumab showed, durable long-term survival effects, something like 45% survival at 1 year, maybe 23–24% at 2 years. Unheard of before.
Wow. That's incredible progress.
And this is a really big but this breakthrough came with a significant cost. A surprisingly high number of patients, around 60%, experienced serious immune-related adverse events, or IRAEs.
60%? That's a lot.
It is. The immune system, once unleashed, sometimes started attacking healthy tissues. This really highlighted a limitation before. The preclinical models hadn't fully predicted this level of human toxicity. It was a true double-edged sword. Unprecedented survival, but really significant, sometimes severe side effects.
Okay, that's where it gets really interesting, doesn't it? Amazing results, but this major toxicity issue. So that must have spurred researchers on, right? To find a more nuanced approach, maybe target a different break with a hopefully better safety profile.
Exactly right. The field knew they needed other options, and the focus then shifted to another crucial brake molecule,
Yeah.
PD-1, programmed cell death protein 1.
PD-1.
PD-1 is another checkpoint, another brake, but it tends to operate more out in the peripheral tissues, like in the organs, within the tumor microenvironment itself, also playing a key role in preventing autoimmunity there.
So CTLA-4 is more like an early break during T-cell activation, and PD-1 is more like a break applied later, out where the fight is happening.
That's a good way to think about it, yes. PD-1 is expressed on activated T cells, but also other immune cells like B cells and NK cells. When it interacts with its partners, its ligand is called PDL1 or PDL2.
PD-L1, I hear that one a lot.
You do, because it's often expressed directly by the tumor cells themselves or by other cells around the tumor. When PD-1 on the T cell binds to PDL1 on the tumor cell, it effectively tells the immune cell, stand down, nothing to see here.
So the tumor puts up its own do not disturb sign directly onto the T cell.
Because that's a major mechanism of tumor immune evasion.
Yeah.
It really shuts down the attack right at the site.
So blocking PD-1 itself or blocking its partner PDL1 would prevent that stand down signal. The tumor couldn't tell the T cells to back off anymore. What were the results this time? And crucially, was the toxicity different from the CTLA-4 blockers?
The results were, again, incredibly impressive. Anti-PD-1 antibodies like nivolumab and pembrolizumab, and then also anti-PD-L1 antibodies like itolizumab, avelumab, durvalumab, they demonstrated potent and, importantly, durable anti-tumor activity.
And in more cancers this time.
Yes, across a much broader range of cancers, not just melanoma, but also non-small cell lung cancer, renal cell carcinoma, bladder cancer, head and neck cancer, Hodgkin lymphoma, colorectal cancer. The list became quite long. And regarding toxicity, while side effects, IRAEs still occurred and could be serious. They were generally less frequent and less severe compared to CTLA-4 blockade. Still higher than predicted by the early models, but often more manageable.
So a wider application and a generally better safety profile, relatively speaking.
Correct. And this widespread clinical success is why you see these anti-PD-1 and anti-PD-L1 drugs approved for so many different cancer types today. They really became a cornerstone of modern cancer therapy for many patients.
The scale of this impact is just incredible. It feels like we went from 0 to 60 very quickly in this field. So what does this mean for the latest developments? Is it just about CTLA-4 and PD-1, or are there other breaks, other pathways being explored, maybe even ways to step on the gas pedal for the immune system?
Well, absolutely. The research hasn't stopped there at all. It's constantly evolving. Beyond CTLA-4 and PD-1, scientists are discovering and revisiting many other checkpoint molecules.
More breaks.
Mostly, yes. More negative regulators. Molecules like LA3, TIN3, TIGIT, VISTA. These are mostly additional breaks that cancer can potentially use, and blocking them is showing promise.
Any examples of those in action?
Sure. For instance, the Relativity 047 trial showed that blocking LG3 along with PD-1 improved progression-free survival in patients with metastatic melanoma compared to just blocking PD-1 alone. That led to an FDA approval.
Combination therapy again.
Exactly. And another one, TIGIT. Blocking TIGIT with a drug called tiragolumab combined with an anti-PD-L1 antibody for metastatic lung cancer actually received FDA breakthrough therapy designation because the combination significantly improved response rates in early trials. So these new targets are definitely moving forward.
It really is like peeling back an onion, finding more and more layers of immune regulation. But you mentioned gas pedals. Are there ways to actively boost the immune response, not just release the brakes?
Indeed. We're also exploring checkpoint molecules that have positive immunoregulatory effects. Think of molecules like ICOS, GITR, OX40. These act more like accelerators or gas pedals for the immune system.
So instead of blocking a brake, you'd want to activate these.
Precisely. The idea is to use agonist antibodies that stimulate or activate these receptors to directly boost anti-tumor immunity. This is a really fascinating area looking at novel therapeutic strategies, although the exact mechanisms and the best ways to use these agonists are still being actively researched and described.
Okay, let's try and quickly recap this first big chunk on the basics of immune checkpoints. It's a lot to take in.
Definitely.
So we've learned about these natural brakes on our immune system checkpoints like CTLA-4 and PD-1. Cancer very cleverly exploits these to hide and evade attack.
Right.
Blocking CTLA-4 with drugs like ipilimumab was revolutionary, especially for melanoma, but came with quite high rates of immune side effects, those IREs.
Yeah, around 60% serious events.
Then, blocking PD-1 or its partner PD-L1, using drugs like nivolumab, pembrolizumab, atezolizumab, offered broader success across many more cancer types, and generally with less, though still significant, toxicity.
A major step forward in terms of applicability and safety profile.
And the research certainly isn't stopping there. Scientists are exploring a whole new family of checkpoints, both inhibitory ones to block like LG3 and TIGIT, and stimulatory ones, the gas pedals like ICOS and OX40, to potentially activate.
That sums it up well, maybe in bullet points for clarity.
Good idea.
So first, immune checkpoints are natural brakes on immunity, but cancer hijacks them. Second, CTLA-4 blockade, like ipilimumab, was the first big win, amazing survival benefits, but high IREs. Third, PD-1, PD-L1 blockade, nivolumab, pembrolizumab, etc., brought broader success. Many cancers were generally less toxic than anti-CTLA-4. And fourth, emerging targets like LAD3, T-shape, more breaks in ICOS, OX40 gas pedals are showing promise, often in combination therapies.
Perfect. That really sets the stage. Now, thinking about all these different brakes and accelerators, it just makes me wonder, how do doctors even begin to decide which one or which combination to use for a specific patient? The mechanism seems so complex.
That is the perfect question, and it leads us directly into our next big segment, because you're right, it's not straightforward.
We know ICB is incredibly powerful. We've just heard about the successes, but it absolutely does not work for everyone. Why? Why is there such variability? What makes some patients respond brilliantly while others, well, others unfortunately resist the treatment?
This is where we really need to dive into the intricate factors at play. We need to look inside the patient at the tumor itself, its genetics, its environment, but also consider broader influences.
Okay, so what are the main categories we're looking at?
It's really a multidimensional puzzle, but we can broadly categorize these factors into, let's say, host-intrinsic factors, meaning things inherent to the patient and their tumor.
Inside the patient.
Right. And then host-extrinsic factors, which we often group under the term exposome, basically. Environmental influences accumulated over a lifetime.
Exposome. We'll definitely need to unpack that later. Let's start with the host-intrinsic factors and maybe begin right at the heart of it, the tumor itself. How does its own DNA, its genetic makeup, influence whether ICB is likely to work?
This is a really fascinating area, centered on the concept of neoantigens.
Neoantigens. New antigens.
Exactly. As tumors grow and divide, they accumulate mutations, errors in their DNA code. Sometimes these mutations change the proteins the tumor cells make, creating unique mutated protein fragments or peptides. These are the neoantigens.
Okay.
Think of these neoantigens as novel wanted posters that the immune system can potentially recognize as foreign or abnormal. They have specific faces that make the cancer stand out.
So the immune system can spot them better.
Potentially, yes. And it turns out that a higher tumor mutational burden, or TMB, meaning the tumor has more of these unique mutations, often correlates with a stronger response to ICB. We see this quite clearly in cancers like melanoma, lung cancer, and bladder cancer.
So in simple terms, the more genetically weird or different the cancer looks compared to normal cells, the better the immune system can target it when the brakes are released.
That's a good way to put it. More mutations often mean more potential wanted posters for the T cells to find. For example, studies show that melanoma patients who had durable, long-lasting responses to ipilimumab tended to have a higher TMB in their tumors.
Okay, so high TMB is often a good sign for ICB response. What about specific types of genetic defects? You hear about things like DNA repair problems. Do they play a role here?
Absolutely. Certain genetic defects are particularly important. Specifically, problems in the DNA mismatch repair system known as DMMR, or a related phenomenon called microsatellite instability, or MSI.
DMMR and MSI. Heard those terms too.
Right. Tumors with these defects are basically faulty at fixing DNA errors. As a result, they accumulate mutations at an incredibly high rate, leading to a very, very high TMB and loads of neoantigens.
So they look really foreign to the immune system.
Extremely foreign. And this makes these DMMR or MSI high tumors highly susceptible to ICB. It's such a strong predictor, in fact, that the FDA granted a landmark tissue-agnostic approval for pembrolizumab.
Tissue-agnostic, meaning?
Meaning the drug is approved for any solid tumor that has this specific genetic feature, DMMR, MSI high, regardless of where in the body the cancer originally started. A major shift towards treating cancer based on its molecular profile, not just its location.
That's incredible. But is TMB the whole story, or can things still go wrong even if a tumor has lots of mutations?
It's definitely not the only story. TMB is a useful biomarker, but it's not perfect. Sometimes you can have a high TMB, but if the tumor have other problems, like defects in the machinery needed to actually present those neoantigen-wanted posters on its surface, well, the T cells still can't see them effectively.
Ah, so the presentation matters just as much as the poster itself.
Precisely. The landscape and quality of neoantigens and the tumor's ability to display them can be just as important, if not more so, than the sheer number of mutations. Defects in antigen presentation pathways are a known mechanism of resistance, even in high TMB tumors.
Okay, moving beyond the raw genetic code and mutations, what about the internal communication pathways within the tumor cells? Do these cellular signals affect how the tumor interacts with the immune system and responds to treatment?
Yes, profoundly. Cancer cells often have abnormal signaling pathways driving their growth. These are called oncogenic signaling pathways. And these pathways don't just control growth, they can dramatically influence the immune response too.
How so?
Well, take pathways like the PI3K pathway or the 1-beta-catenin pathway. Activating mutations in the PI3K pathway, for instance, can cause tumor cells to ramp up their expression of PD-L1.
The do not disturb sign again.
Exactly. They put up a stronger shield against T cells.
Yeah.
Activation of the Wnt-beta-catenin pathway, on the other hand, have been linked to hindering the recruitment of immune cells into the tumor microenvironment in the first place. It creates an immune-excluded state.
So these pathways can either make the tumor harder to attack directly or stop the attackers from even getting close.
Correct. And understanding this complex internal crosstalk is really important. It often forms the basis for developing combination therapies.
Ah, like adding a drug that blocks the PI3K pathway alongside an ICB drug.
Precisely. The idea is to block those resistance-driving signals within the tumor cell at the same time as you're releasing the brakes on the T cells, hoping for a synergistic effect, a better overall response.
This makes sense. It sounds like the tumor isn't just sitting there passively, it's actively strategizing. Is it even sending out messages like little drones or smoke signals to suppress the immune system further away?
That's actually a surprisingly accurate analogy. Recent studies have shown that tumors release tiny packages called extracellular vesicles, or EVs. A specific type called exosomes are particularly interesting.
Exosomes, tiny messengers.
Exactly. And what's really fascinating and concerning is that some of these tumor-derived exosomes carry functional PD-L1 right there on their surface.
Wait, PD-L1 on these tiny packages floating around?
Yes. So these mobile PD-L1 molecules can travel through the body and suppress CD8 plus T cells far away from the actual tumor site, maybe even in the lymph nodes where T cells get activated. They can induce T cell exhaustion systemically and promote tumor growth.
That's insidious, like remote jamming of the immune response.
It is. And clinically, higher levels of these circulating exosomal PD-L1s found in a patient's blood before starting treatment have been linked to a lack of benefit from ICB. This makes them a really promising potential biomarker to predict who might not respond well.
Wow. Okay, so we've talked about the tumor's genetics, its internal signaling, even these messenger vesicles. But it's not just the tumor cells in isolation, right? What about the surrounding neighborhood, the tumor microenvironment, or TME?
Absolutely critical. The TME is a whole ecosystem. It's not just cancer cells. It includes various non-immune stromal components.
Stromal components, like what?
Things like endothelial cells that form blood vessels and cells called fibroblasts that provide structural support.
And how do they influence things?
Well, the blood vessels in tumors are often abnormal, a process called angiogenesis. These leaky, disorganized vessels can act like a physical barrier, literally impairing the ability of immune cells to infiltrate the tumor and also affecting how well ICB drugs can penetrate.
Like a poorly built road network stopping the troops getting in.
Exactly. Plus, the endothelial cells themselves can express PD-L1, directly putting the brakes on T cells trying to exit the bloodstream into the tumor.
Okay, what about the fibroblasts?
Cancer-associated fibroblasts, or CHEFs, are really complex. They can have dual effects. Some types might actually help create a more immune-inflamed or hot tumor by secreting certain factors. But other types, like those expressing a marker called FAK, actively inhibit T cell function and promote resistance to ICB.
So even the support cells can be friend or foe.
Very much so. And it can be tissue-specific, too. For example, in bone metastases, specialized cells called osteoclasts can release factors like TGF-beta, which reduces anti-tumor T cells and drives resistance specifically in that environment. The local neighborhood really matters.
Which brings us, of course, to the immune cells within that neighborhood. They must play a huge role. I've heard people talk about hot and cold tumors in this context. What does that actually mean?
Yeah, that's a really crucial distinction. Gets at the question, is the immune system already engaged with the tumor or not? So immune-inflamed or hot tumors are basically those where the immune system has already infiltrated. You see abundant CD4 plus and CD8 plus T cells right within the tumor mass. These tumors, generally speaking, tend to respond much better to ICB. The soldiers are already there, they just need the brakes released.
Makes sense. And cold tumors.
Cold tumors are the opposite. They fall into a couple of categories. Immune-excluded tumors might have T cells nearby, maybe stuck in the surrounding stroma, but they haven't actually penetrated the tumor mass itself. Then you have immune desert tumors, where there are basically very few or no T cells around at all.
And these cold tumors are generally resistant.
Typically, yes. If the T cells aren't there or can't get in, releasing the brakes doesn't help much. And unfortunately, as tumors evolve, they can often manipulate their microenvironment to become more immunosuppressive, essentially turning a potentially hot tumor cold over time.
So if the CD8 plus T cells are the main soldiers we want attacking the tumor, who are the saboteurs within this microenvironment? Which immune cells are actively suppressing the attack in these cold or resistant tumors?
There are several key players acting as saboteurs. We already mentioned regulatory T cells, or TREGs.
Right, boosted by CTLA4.
Exactly. They normally maintain self-tolerance, but in the TME, they actively suppress the anti-tumor T cells through various mechanisms, including expressing checkpoints like CTLA4 and PD-1 themselves. While ICB can sometimes deplete them, they can also become resistant.
Okay, who else?
Another important group are myeloid-derived suppressor cells, or MDSCs. These are immature myeloid cells with potent immunosuppressive activity. They often express PD-L1, contributing to T cell suppression, and lower numbers of MDSCs have been linked to better responses to drugs like ipilimumab.
MDSCs, another suppressor cell type.
And then there are tumor-associated macrophages, or TAMs. These are particularly interesting because they're very plastic. They can change their function, meaning they can swing between two main types. M1 macrophages are generally pro-inflammatory and help fight the tumor. But M2 macrophages are anti-inflammatory, immunosuppressive, and actively promote tumor evasion and growth. In many resistant tumors, the M2 type dominates.
So you'd want to shift the balance from M2 back to M1.
Exactly.
Yep.
Strategies to inhibit M2 TAMs or reprogram them towards an M1 phenotype are actively being explored as ways to enhance ICB efficacy. It's about changing the character of the neighborhood guards.
TREGs, MDSCs, M2 macrophages, quite a gang of suppressors. Are there any immune cells in the TME that are surprisingly helpful?
Yes, actually. B cells, which are typically known for making antibodies. When B cells organize with T cells into structures called tertiary lymphoid structures, or TLS, right within or near the tumor.
TLS, like mini lymph nodes in the tumor?
Kind of, yeah. The presence of these TLS structures has been associated with better responses to ICB in several cancer types. The B cells within them might be helping to activate T cells or even producing anti-tumor antibodies. So B cells can sometimes be allies in this fight.
So it's not just the tumor cells themselves, but this whole intricate ecosystem of their genetics, their signaling, their messengers, the surrounding stromal cells, and this diverse cast of immune cell suppressors and potential helpers, all interacting in the local microenvironment. It really is a deep dive into tumor microecology.
It truly is. And we're not even quite done with the intrinsic factors. Beyond the cells, the overall metabolic state of the microenvironment plays a role.
Metabolism, like energy use.
Yeah. For instance, tumors often have areas with low oxygen called hypoxia. Hypoxic conditions can decrease the tumor's ability to present those neoantigen-wanted posters, making it harder for T cells to see them. Dysregulated nutrient availability also affects immune cell function.
Okay, metabolism matters too. Anything else in this local environment?
One really emerging and, I think, underappreciated component is the intratumoral microbiome.
Wait, microbes inside the tumor itself? Bacteria living with the cancer cells?
Yes. It sounds strange, but researchers are characterizing bacterial communities that reside within human tumors. And these aren't just passive bystanders.
How could they possibly influence things?
Well, the types of bacteria found can be specific to the tumor type. They seem to influence tumor growth, modulate the local immune microenvironment, and potentially even affect response to therapies, including ICB.
Wow, how?
One fascinating finding is that peptides, small protein fragments derived from these intratumoral bacteria, can actually be presented by the tumor cells on their surface, just like neoantigens. And T cells can recognize these bacterial peptides.
So the key cells might be reacting to the bacteria in the tumor.
It seems plausible, yes. It adds another layer of complexity to what the immune system is seeing and responding to. And intriguingly, differences in the tumor microbiota composition have been observed between melanoma patients who respond to ICB and those who don't.
Mind blown. Bacteria inside tumors affecting immunotherapy. Okay, this section on host-intrinsic factors has been incredibly detailed. Let's try to quickly summarize the key points before we move on.
Good idea. It's a lot of interconnected pieces.
So we started with the tumor's own genetics. High tumor mutational burden, TMB, often driven by defects like DMMR or MSI, creates more neoantigens, those wanted posters, making the tumor more visible to the immune system and predicting better ICB response.
Right. Genetics and neoantigens are foundational.
Then we looked at the tumor's internal signaling pathways like PI3K or WNT beta-catenin, which can help the tumor hide by increasing PD-L1 expression or blocking immune cell entry.
Key resistance mechanisms originating within the cancer cell.
And tumors can even send out tiny messenger vesicles, exosomes, carrying PD-L1 to suppress T cells remotely.
Those circulating exosomes are important signals.
Then we moved to the tumor microenvironment, the TME. The stromal blood vessels and fibroblasts can act as barriers or suppress immunity.
The physical and cellular neighborhood matters.
And within that neighborhood, the immune cells are crucial. Hot tumors, full of CD8 plus T cells, respond better than cold tumors. Suppressor cells like Tregs, MDSCs, and M2-type macrophages actively dampen the immune attack in resistant tumors.
The immune cell balance is critical. Hot versus cold.
And finally, even the local metabolic conditions like hypoxia and the presence of an intratumoral microbiome bacteria within the tumor can significantly impact anti-tumor immunity and ICB response.
Don't forget the local metabolism and microbes.
So the overarching goal seems to be understanding all these factors to figure out how to turn cold-resistant tumors hot and more responsive to treatment.
Exactly. Addressing these intrinsic mechanisms of resistance is a major focus of current research.
Okay, summarizing those key takeaways again, maybe bullet style.
Sure. Tumors genetics. High TMB, DMMR, MSI status strongly predict response due to neoantigens. Tumor signaling. Pathways like PI3KWMAnt can drive resistance, usually via PD-L1 or immune exclusion. Extracellular vesicles, tumor exosomes carrying PD-L1 suppress T cells systemically. Tumor microenvironment, TME. Stromal cells, vessels, CFFs, and immune cells. Tregs, MDSCs, M2-TAMs, create suppressive cold environments, while hot infiltrated tumors respond better. Metabolism and microbes. Local hypoxia, metabolism, and even bacteria within the tumor influence immunity and ICB outcomes.
Fantastic summary. Now, given all these incredibly complex internal factors happening right at the tumor site, I'm wondering, what's maybe one surprising way the patient's overall health, their whole system, or even things from their environment might contribute to whether ICB works or fails?
That's a perfect segue into the next layer of complexity. Because you're absolutely right. Cancer immunity isn't purely a local phenomenon.
That's what I was thinking. It takes us beyond the tumor and its immediate neighborhood, right? To the patient's whole system and maybe even their lifetime of environmental exposures. How do these host systemic factors and this exposome thing play into ICB response and, just as importantly, the side effects?
Exactly. We need to zoom out. Cancer immunity is systemic. For instance, something as fundamental as our human leukocyte antigen, or HLA genotype, plays a role.
HLA, that's involved in presenting antigens, right? Like showing the wanted posters.
Precisely. HLA molecules are what display those neoantigens and other antigens to T cells. The diversity of your HLA genes dictates how wide a range of antigens your body can effectively present.
So more diversity is better?
Generally, yes. Studies have shown that individuals who are homozygous at certain HLA loci, meaning they inherited identical copies from both parents, resulting in less diversity in antigen presentation, tend to have decreased survival when treated with ICB compared to those with more diverse HLA types.
Interesting. So your inherited immune system display case matters. What other systemic factors are important? I feel like I hear a lot about the gut microbiome these days.
You absolutely do, and for good reason. Our gut microbiome, the trillions of bacteria and other microbes living in our intestines, is absolutely essential. Think of it as a crucial immune training ground.
Training ground, how so?
These gut microbes constantly interact with our immune system from birth, helping it learn to differentiate between friend and foe, tuning its responses, and there's now compelling evidence linking the composition of a patient's gut bacteria directly to how well they respond to ICB.
Really? In which cancers?
It's been shown in preclinical models, but importantly also in patients with melanoma, renal cell carcinoma, and non-small cell lung cancer, among others. Specific bacterial species seem to be associated with better responses, while others are linked to resistance.
That's incredible. Does that mean things affecting our gut bacteria could impact cancer treatment? Like antibiotics?
Yes, and that's a critical point. Studies have shown that treatment with common broad-spectrum antibiotics, which can wipe out gut microbial diversity, can negatively impact ICB outcomes in patients. It really underscores how interconnected things are.
Wow. So conversely, could things that promote gut health help ICB?
The evidence is pointing that way. Things like a high-fiber diet, which feeds beneficial gut bacteria and leads to the production of things like short-chain fatty acids, have been linked to improved ICB response and survival in some studies. Exercise also seems to promote beneficial gut diversity.
So diet and exercise could potentially influence immunotherapy response through the gut. Are people trying to manipulate the microbiome therapeutically?
Yes, very actively. Clinical trials are exploring strategies like fecal microbial transplantation (FMT), where gut microbes from a healthy responding donor are transferred to a non-responding patient. Others are testing specific probiotics or prebiotics to try and modulate the gut microbiome in beneficial ways during ICB treatment. It's a very hot area of research.
Fascinating. Gut health impacting cancer therapy. What about other systemic factors? Our overall health, maybe things like weight or hormones?
Also relevant. What's really interesting and somewhat paradoxical is the finding regarding obesity.
Paradoxical how? Usually obesity is linked to worse cancer outcomes.
Generally, yes. But with ICB, particularly in certain cancers like melanoma and NSCLC, several studies have surprisingly found that patients with obesity, defined as BMI over 30, actually had improved responses in survival compared to non-obese patients. This effect seems more pronounced in men.
Better outcomes with obesity. That seems counterintuitive. Do we know why?
The exact reasons are still being investigated. It might involve altered immune cell function, inflammation levels, or metabolic changes associated with obesity that somehow synergize with ICB in some contexts. It's complex.
And you mentioned men potentially responding differently. Do sex hormones play a role?
It appears so. There are sex-based differences in immune responses generally, and some data suggest men might show better ICD responses in certain situations, possibly linked to hormonal influences on things like PD-L1 expression. This has even prompted clinical trials looking at combining ICB with anti-estrogen or anti-androgen therapies.
So our inherited immune genes, HLA, our gut bacteria, our weight, our hormones, all these systemic factors intrinsic to the patient matter. Now, what about the other category you mentioned, the exposome? That sounds big.
It is big. The exposome is formally defined as the cumulative measure of environmental influences and associated biological responses throughout the lifespan.
Okay, cumulative lifespan. So basically everything non-genetic that happens to us.
Pretty much, yeah. It encompasses a huge range of factors. Where you live, your diet, infections you've had, medications you take, exposure to pollutants or radiation, cosmetics you use, psychosocial factors like chronic stress and depression or anxiety.
Wow, stress and anxiety too.
Yes. Also, broader factors like socioeconomic status, access to healthcare and nutritious food, even things like climate change, and systemic issues like racial injustice can be considered part of the exposome's influence on health.
That's incredibly broad. How do you even begin to study something so vast and dynamic that builds up over a whole life?
It's a major challenge, methodologically. But researchers are developing tools and approaches, and we're already seeing inklings of how specific exposome factors can impact tumor immunity and ICB response.
Like what? Give me an example.
Okay, think about UV radiation from sunlight or exposure to cigarette smoke. Both are known carcinogens, but they also increase the mutation rate in skin cells and lung cells, respectively.
Leading to higher TMB and more neoantigens.
Exactly. This helps explain, in part, why melanoma and non-small cell lung cancer, cancers heavily influenced by these environmental exposures, often have relatively high TMB and consequently relatively high response rates to ICB compared to some other cancers. The exposure history shaped the tumor's immunogenicity.
That makes sense. What about stress? You mentioned that.
Chronic stress is known to have immunosuppressive effects through various hormonal and neural pathways. It can potentially dampen anti-tumor immune responses. Interestingly, on the flip side, some retrospective data suggested that patients taking non-selective beta blockers, drugs often used for high blood pressure or anxiety, which block certain stress hormone receptors, had improved overall survival when treated with ICB.
Really? Taking beta blockers improved ICB outcomes?
The retrospective data suggested a correlation, yes. This has prompted prospective clinical studies to investigate this properly. It hints that modulating stress pathways might be beneficial.
Fascinating. Are other medical treatments considered part of the exposome in this context?
Definitely. Prior or concurrent cancer treatments like chemotherapy and radiation therapy can be viewed as part of a patient's relevant exposure history. And they don't just kill cancer cells, they can also have significant immune modulatory effects.
How so?
Some chemotherapies can induce a type of cell death that's more immunogenic, releasing tumor antigens. Radiation can increase PD-L1 expression on tumor cells, potentially sensitizing them to subsequent PD-1 blockade. It could also enhance antigen presentation. This is why combination strategies involving chemo or radiation with ICB are often very effective. The prior treatment is part of the exposure that primes the system.
So it truly isn't just about the ICB drug itself, but the entire context of a patient's life, their genetics, their systemic health, their microbiome, and this vast accumulation of environmental and lifestyle exposures, past and present, that all feed into whether the treatment works. This is a really profound shift in understanding cancer therapy.
It absolutely is. It's moving towards a much more holistic and personalized view.
Okay, we've talked a lot about what makes ICB work or not work. Now we need to turn to the other crucial side of the coin, the toxicity. We touched on it with CTLA-4, but let's delve deeper. What are these immune-related adverse events, the IRAEs, actually like? And how common are they really when we unleash the immune system this powerfully?
IRAEs are, without doubt, a major challenge and a key limiting factor for ICB. They represent the immune system, hyperactivated by the therapy, mistakenly attacking healthy tissues.
And they can affect anywhere.
Pretty much any organ system. The skin, rashes, vitiligo, the gastrointestinal tract, colitis, diarrhea, the lungs, pneumonitis, the endocrine glands, thyroiditis, hypophysitis, the liver, hepatitis, the joints, arthritis, the nervous system, the heart, myocarditis.
Yeah.
Over 70 different specific pathologies have been documented. It's incredibly diverse.
70 different conditions. That's staggering. How severe do they get?
The severity varies widely. Many patients experience low-grade effects, maybe grade 1 or 2, which can be managed. These might occur in up to 90% of patients, depending on the drug or combination. But more severe effects, grades 3 to 5, which can be life-threatening or fatal, are also seen, maybe ranging from 20% up to 60% of patients, particularly with combination therapies like anti-CTLA-4 plus anti-PD-1.
And when do they happen? Is it predictable?
That's another challenge. Their onset is highly unpredictable. Some IRAEs can occur within days of starting treatment. Others might not appear for months, or even after treatment has finished. And the pattern varies between different ICB agents and combinations. It requires constant vigilance.
How are they typically managed when they do occur?
The standard approach for moderate to severe IRAEs involves stopping the ICB treatment, at least temporarily, and administering high-dose corticosteroids, like prednisone, to suppress the immune system.
Steroids. But don't they counteract the anti-cancer effect we want?
That's the concern, and we'll come back to that. Managing severe IRAEs often requires hospitalization and input from a multidisciplinary team of specialists, oncologists, gastroenterologists, endocrinologists, dermatologists, etc., depending on the organ affected.
It sounds incredibly complex to manage. But why do these IRAEs happen? What's the underlying mechanism? Is it simply the immune system going haywire and attacking self?
That's the million-dollar question, and honestly, we lack a full, complete mechanistic understanding. Several possibilities are proposed, and they likely aren't mutually exclusive. Different mechanisms might drive different types of IRAEs.
Okay, what are some of the leading theories?
One idea is on-target, off-tumor effects. This suggests T cells activated against a tumor antigen might recognize a similar-looking antigen that's also present on normal, healthy tissue. So the T cell attacks both. This might explain things like myocarditis, heart inflammation, or certain rashes where short antigens are known.
The T cell hits the right target but on the wrong cell type.
Exactly. Another theory is epitope spread. As the ICB kills tumor cells, these dying cells release a whole bunch of previously hidden tumor antigens. The immune system mounts a response against these new antigens, but some of them might again resemble self-antigens found on healthy tissues, leading to autoimmunity.
So the immune response broadens, but sometimes too broadly.
Right. A third possibility involves pre-existing autoreactive T cells. We all have some T cells capable of recognizing self-antigens, but they're normally kept dormant or suppressed by those very immune checkpoints we're blocking. ICB might release the brakes on these pre-existing autoreactive cells, allowing them to attack healthy tissue.
Unleashing sleeping dogs, essentially.
Sort of, yes. And it's not just T cells. The humoral immune system, B cells and antibodies, are also implicated. Early changes in B cell populations have been associated with developing toxicity. Other immune cells, like myeloid cells, might contribute to the inflammation. And in some cases, like hypophysitis, pituitary gland inflammation with anti-CTLA-4, it might even involve the antibody drug directly binding to CTLA-4 expressed on the pituitary gland itself, causing damage.
So, multiple potential mechanisms driving these diverse side effects. Is there any link between getting an IRAE and having a good response to the cancer treatment? Like, is it a sign the immune system is really fired up?
There's mixed data on that. Some retrospective studies have suggested a correlation. Patients who develop certain IRAEs, like skin toxicities or endocrine problems, sometimes seem to have better anti-tumor responses. For example, a genetic predisposition score for vitiligo, skin depigmentation, was found to predict ICB response in bladder cancer.
Interesting, but
Yeah.
not always the case.
Not consistently, no. And it's confounded by the treatment for the IRAEs themselves. The critical question is, do the high-dose corticosteroids given to treat the IRAEs then detrimentally impact the anti-tumor response we want?
Right, the potential trade-off.
The answer likely depends on the timing, dose, and duration of the steroid treatment. Some studies suggest that early use of high-dose steroids, especially for prolonged periods, might be associated with decreased overall survival in patients receiving ICB. It's a real balancing act. Managing toxicity without sacrificing efficacy is a huge ongoing challenge, especially since patients who experience severe IRAEs might have limited options for receiving further immunotherapy in the future.
A very difficult tightrope to walk for doctors and patients. Okay, so with all this incredible complexity, all these host factors, tumor factors, exposome factors influencing response, and this whole spectrum of unpredictable toxicities, how are scientists trying to get smarter about this? How can we better predict who will benefit and who will get severe side effects?
Precision medicine is the goal. We need better tools to predict both response and toxicity before starting treatment or very early on.
What kind of tools, what biomarkers are showing promise?
For predicting response, we have some established genetic biomarkers like TMB, MSI status, and DMMR status. As we discussed, these led to those groundbreaking tissue-agnostic FDA approvals for pembrolizumab. They indicate a high likelihood of response.
Okay, TMB and MSI DMMR. What else?
Then there are immunological biomarkers, looking at the presence and type of tumor-infiltrating lymphocytes, TILs, especially CD8 plus T cells, within the tumor biopsy can be informative. More T cells often means better response, hot tumors. PD-L1 expression level on tumor cells or immune cells is also widely used as a biomarker for some ICB drugs, although its predictive value can be inconsistent across different cancers and assays.
Right, PD-L1 testing is common but not perfect. Are there newer strategies emerging?
Definitely. Researchers are looking at more sophisticated analyses of T cell markers and function within the tumor. Remember those circulating exosomal PD-L1s we talked about? Combining their levels with other markers might improve prediction. And critically, microbiome signatures analyzing the bacteria in both the gut, from stool samples, and even within the tumor biopsy, or microbes circulating in the blood are emerging as powerful potential predictive biomarkers for response.
Microbiome signatures. That's cutting edge. What about predicting toxicity, the IRAEs? That seems just as important.
Absolutely. Predicting toxicity is perhaps even less developed, but equally crucial. Potential biomarkers for IRAEs are being investigated. These include certain HLA genotypes. Some HLA types are linked to specific autoimmune diseases, signs of gut microbiota dysbiosis, imbalance before treatment, features extracted from radiological scans using AI, radiomics, and changes in peripheral blood immune cell signatures, for example, looking at T cell repertoire diversity, levels of specific inflammatory cytokines like IL-17 or IL-6, or certain B cell subsets like CD21 low B cells.
So looking for pre-existing immune signatures that might flag a risk for autoimmunity once the brakes are off.
Exactly. The hope is to eventually combine multiple biomarkers, genetic, immunological, microbial, clinical, into algorithms that can give a much better personalized prediction of both benefit and risk for each patient.
That makes sense. Now, beyond just predicting response and toxicity, how are we actively trying to improve the treatments themselves? How can we boost response rates, overcome resistance, and make these therapies work for more people?
That's where therapeutic strategies come in, building directly on our understanding of those resistance mechanisms we discussed earlier.
Okay, like what?
Well, since we know epigenetic changes can silence antigen presentation machinery, researchers are testing combinations of ICB with epigenetic modulators, like DNA methyltransferase inhibitors, to try and make tumors more visible to the immune system again.
Undoing the silencing.
Right. We talked about aberrant signaling pathways driving resistance. So combining ICB with targeted signaling modulators is a major strategy. The combination of BRAF inhibitors, which target a common mutation pathway in melanoma, with anti-PD-L1 antibodies, is already standard care in melanoma, showing much better results than either approach alone.
Targeting the cancer's internal wiring and the immune breaks.
Precisely. Another approach uses cytokines, which are immune signaling molecules. Next-generation versions of cytokines like IL-2 or IL-12 are being engineered to hopefully boost the number and function of tumor-infiltrating lymphocytes, TILs, more effectively and with less systemic toxicity than the older versions.
Boosting the soldiers. What about the tumor neighborhood, the TME?
We can target that too. Anti-angiogenic agents, which normalize tumor blood vessels, can be combined with ICB to improve immune cell infiltration. The combination of bevacizumab, an anti-angiogenic, plus atezolizumab, anti-PD-L1, is now FDA approved for liver cancer, for example.
Fixing the road so the troops can get in.
Good analogy. And as we mentioned, conventional chemotherapy and radiotherapy can have immune modulatory effects, so combining them rationally with ICB is a very common and often successful strategy across many cancers.
Leveraging the old tools in new ways.
Exactly. Plus, there are newer approaches like using oncolytic viruses engineered to selectively infect and kill cancer cells, while also stimulating an anti-tumor immune response. Or developing therapeutic cancer vaccines designed to expand the pool of tumor-specific T cells. All these strategies aim to overcome resistance, often by trying to turn those cold tumors hotter and maximizing the immune attack.
A whole arsenal of combination strategies. Now, critically, what about the other side of that tightrope? What strategies are being developed to specifically make ICB safer? To prevent or better treat those challenging IREs and minimize the collateral damage?
This is hugely important. We're definitely moving beyond just relying on generalized high-dose corticosteroids for every IRE.
More targeted approaches for toxicity.
Yes. For example, for dermatologic IREs, topical steroids might be sufficient. For severe colitis, gut inflammation that doesn't respond to steroids, more specific drugs targeting inflammatory pathways, like TNF inhibitors, for example, infliximab, or drugs targeting gut-specific immune cell trafficking, like vedolizumab, an alpha-fosfolbeta-7 integrin inhibitor, are showing success.
Drugs specific to the type of inflammation.
Right. And remember the gut microbiome's role. There have even been case reports in small studies where fecal microbiota transplantation FMT has been used successfully to treat refractory steroid-resistant colitis caused by ICB.
Using gut bacteria to treat gut inflammation caused by immunotherapy. Fascinating.
It really is. But perhaps the most exciting area for improving safety lies in engineering improved immunotherapeutic agents themselves, making the drugs inherently safer.
How can you do that?
One major focus is on developing bispecific and trispecific antibodies. These are synthetic engineered antibodies designed to bind to 2 or even 3 different targets simultaneously.
2 or 3 targets at once. How does that help safety?
The idea is to create more targeted effects. For example, you could design a bispecific antibody that only strongly activates a T cell via a costimulatory molecule like CD137 if it also recognizes a tumor-specific antigen. This could focus the T-cell activation much more precisely to the tumor site, reducing off-target activation and toxicity in healthy tissues.
Conditional activation only at the tumor.
Exactly. Or thinking about those agonist antibodies for the gas pedal molecules like OX40, sometimes activating them everywhere can cause liver toxicity. So researchers are designing dual agonistic bispecific antibodies, for instance, targeting both OX40 and CD137, engineered in a way that might boost T-cell activity effectively, but with potentially reduced liver toxicity compared to stimulating just one pathway broadly. It's about engineering precision into the therapeutic molecule itself.
That sounds like the future smarter, more targeted drugs.
Okay, let's try to bring this all together in a final recap. This has been a really deep dive.
It certainly has lots of interconnected layers. So we learned that factors beyond the tumor itself are systemic health, including our unique HLA type, our crucial gut microbiome, influenced by things like antibiotics, diet, exercise, potentially even factors like obesity and sex hormones, can significantly sway ICB outcomes.
Right, the patient's internal systemic environment is key.
Then we explored the vast exposome, that lifetime accumulation of environmental influences, everything from sunlight and smoking history to chronic stress and even prior cancer treatments. These external factors play a hidden but powerful role in shaping tumor immunogenicity and ICB response.
The exposome adds another critical layer of complexity and opportunity.
And while these ICB therapies are truly life-changing for many, we're constantly grappling with the challenge of IRAEs, those unpredictable immune-related side effects where the unleashed immune system can sometimes target healthy tissue, requiring careful management and sometimes difficult trade-offs.
Toxicity management is paramount.
But the field is rapidly advancing. We're developing better predictive biomarkers, genetic, immunological, exosomal, microbial, to try and identify who will benefit most and who is at highest risk of toxicity.
Precision diagnostics are crucial.
And we're designing smarter therapeutic strategies using rational combinations that target tumor signaling, epigenetics, the TME, or leverage chemoradiaviruses vaccines to enhance response and overcome resistance, often aiming to turn cold tumors hot.
Combination therapies are key to improving efficacy.
And finally, we're working on strategies to specifically abrogate toxicity, moving beyond just steroids to use targeted drugs for specific IRAEs, like TNF inhibitors for colitis, and engineering next-generation ICBs, like those clever bispecific antibodies, designed to be more precise and minimize off-target effects from the start.
Engineering safer, more effective therapies is the ultimate goal.
It really feels like we're moving towards a truly personalized future in cancer immunotherapy.
That's definitely the direction. Integrating all this knowledge is key.
Okay, final summary bullets for this last section.
Let's do it. Systemic factors. HLA genotype, gut microbiome affected by diet, antibiotics, obesity paradox, sex hormones all impact ICB. Exposome. Lifetime environmental factors, UV, smoking, stress, other treatments, includes tumor immunity and ICB outcomes. IRAEs. Immune side effects are common, diverse, unpredictable, sometimes severe. Managing them is critical. Predicting and enhancing. Biomarkers, genetic, immune, microbiome, and combination therapies aim to improve response prediction and efficacy. Abrogating toxicity. Targeted IRAE treatments and next-gen engineered antibodies like bispecifics aim to improve safety.
Perfect. That brings us to the end of our deep dive. Knowing all of this, all these intricate connections, what do you see as one major opportunity, maybe the biggest leap forward we might see in the near future for these treatments, given this holistic understanding how everything from our gut bacteria to our stress levels can impact cancer therapy?
That's a great question to ponder. For me, I think the biggest opportunity lies in truly integrating all these diverse streams of data. The genomics, the proteomics, the microbiome, the imaging, the exposome factors, the clinical data, using sophisticated computational approaches, maybe AI.
Integrating everything.
Yes, to create a truly personalized, dynamic risk-benefit profile for each individual patient before treatment even starts, and potentially adapting it over time. This isn't just about picking drug A versus drug B anymore. It's about deeply understanding the entire patient ecosystem, their tumor, their immune system, their environment, their lifestyle, to predict, prevent, and proactively manage both response and toxicity in a way we just haven't been able to before. That, I think, is the next frontier.
A truly holistic, data-driven, personalized approach. That's a powerful vision. This deep dive today has really shown us, I think, just how intimately linked our bodies, our environment, and our response to these powerful treatments truly are. So thinking about that vastness of the exposome, we discussed, everything from your diet, your exercise habits, your stress levels, where you live, what's maybe one unexpected factor in your own daily life that you, our listener, think might be influencing your health and your immune system in ways we're only just beginning to really understand scientifically. After hearing all this, what's one thing you're now curious to maybe explore further?
It's definitely food for thought, and there's always more to learn. Critical thinking, considering multiple perspectives, it always enriches understanding. Keep questioning those assumptions.
Well said. Thank you so much for joining us on this deep dive into the complex world of immune checkpoint blockade. Until next time, keep learning, keep questioning, and stay curious.